Right here we have the cell with its nuclear envelope and 2 chromosomes in them. Each chromosome is made up of chromatin, which is DNA wrapped around histones. It's also applicable to this pink one. And right now it's in the cell is in the G0 phase. Once called upon to enter mitosis, it enters the G1 stage of interphase, which is the first step of mitosis. In G1, the organelles duplicate, more enzymes are made, and more ATP is made to add to mitosis. After G1, synthesis happens. And in synthesis, there is identical copy made of each DNA in the chromosome, as you can see by the same white one here and the same purple here, and then the same thing for these guys. And these 2 pairings are called sister chromatids. So these are sister chromatids and these are sister chromatids. And like I mentioned, they have identical genes of either big B or little b, little w or big W, big G or big or little g, and big P or little p. These are the alleles of each gene, but for now they're just matched up by their genes. After synthesis, we move into G2, which is when the cell preps for division and the chromatin condenses and becomes chromosomes. After G2, we enter into the next phase, out of interphase, prophase. In prophase, the chromatins cross over and create centromeres. Okay, in prophase, we have the centromeres, and we count how many chromosomes are in the cells based on the number of centromeres. So in this example, we have 2 chromosomes because we have 2 centromeres. At this point, the nuclear envelope also disappears. This is different from meiosis because the 2 chromatids are identical and no crossing over occurs. Now we enter into metaphase. All that the chromosomes do is line up in a single file line within the cell. After metaphase, we enter into anaphase. In anaphase, we introduce our centrioles, which are parts of the cells, and we have centrioles host the centromeres, which connect to the spindle fibers that connects to the centromeres of each chromosome or are free to hold the structure of the cell.
Yeah.
These are the spindle fibers. And the spindle fibers will pull apart the chromosomes. At this point of anaphase, the spindle fibers will bring apart the chromosomes towards the centriole. And now where there once was 2 centromeres, there is now 4 centromeres. So now we have 4 chromosomes in the cell. Now we enter into telophase, where the chromosomes are split apart and the nuclear envelope begins to form again, creating the cleavage furrow between the 2 dividing cells. After telophase, we enter the final stage, which is cytokinesis, when the 4 chromosomes get split into 2 cells, each with 2 chromosomes. So we have these guys going around this new cell, and now we have a new cell forming over here as well. Now we have 2 identical somatic cells, which are body cells, such as a knee cell being made for a knee. The end.
Meiosis is the process that creates gamete cells, which are sex cells. These cells are different from somatic body cells that are produced in mitosis, as cells from meiosis are haploid cells, meaning they have 23 chromosomes, 22 that are autosomal and 1 that determines the gender, which differs from diploid cells that have 46 chromosomes, 44 autosomal, and 2 that determine the gender. Meiosis's goal is diversity and to create gametes that are all different, while mitosis creates identical cells. The starting point of meiosis is identical to mitosis, which is interphase 1. In interphase, identical sister chromatid copies are made. Now this is the end of interphase 1. Now each chromosome has its identical sister chromatid copy. Now we will begin prophase 1. In prophase 1, the nuclear membrane begins to disappear. And synapsis occurs, which is the movement of homologous chromosomes which have the same genes together and attach to the centromeres. For example, a chromosome number 5 are attached to another chromosome number 5, each from mom and dad. Each chromosome 5 would contain the same genes, which is why they are able to attach during synapsis. After synapsis, we have now created tetrads, which are 4 sister chromatids attached together at the same centromere. This differs from mitosis because in mitosis there are only 2 sister chromatids attached at a centromere. And after synapsis, the process of crossing over begins. This is when genes of homologous chromosomes are switched. For example, shown here, we can switch big P with little p. And here, you can switch big G with little g. This process creates diversity of genes among each gamete to make them all different. The region where the crossover occurs is the chiasmata. So this is where the chromosomes are touching. We are now in metaphase 1. This process occurs after the crossing over of genes. This is when the homologous chromosome pairs line up next to each other in the middle of the cell. This process differs from mitosis, as mitosis there are only 2 sister chromatids in the middle, not 4. In metaphase, there is a Mendel law called the law of independent assortment, meaning that the chromosomes can be on either side of the cell as long as they stay with their homologous pair. For example, there could be a pair of pink on either side, like this, or they could stay like this, and the orange chromosomes could go on this side. This process is completely random. In this specific example, we have an n value, which means the number of homolog pairs, and 2n, the number of total chromosomes. So our 2n value is 8, and our homolog pair is 4. Our n value is 4. At the end of metaphase, microtubules or spindle fibers come from the centrioles on both sides of the cell and attach themselves to the kinetochores inside each centromere of each homologous pair. This is how we begin anaphase 1. Using the spindle fibers just attached, the centrioles pull apart each chromosome from their homologous pair to opposite poles of the cell. In anaphase, the tetrad, which was the 4 sister chromatids together, is now gone, and the homologs will never see each other again. This is called Mendel's law of segregation. When homologs leave to be a part of a different cell, and they never see each other again, to cross over more genes or to get back the ones they've already crossed. Now this is the process of telophase 1. Now that each pole of the cell has different haploid sets of chromosomes, the cleavage furrow can begin to form, almost dividing the cell into 2. Nuclear membranes begin to reform around the chromosomes. And once they do form around the chromosomes, this is cytokinesis. Cytokinesis is the process of the cell officially becoming 2 different cells. That ends meiosis 1. Now it is time for meiosis 2. This process is very similar to mitosis. Processes are the same, except chromosomes are already copied, so interphase is skipped, and prophase is now condensing the chromosomes, and the nuclear membrane is disappearing once again. Metaphase lines up each chromosome in the middle of the cell, and anaphase attaches the centrioles to each centromere. Now we are in telophase 2, which 4 nuclear membranes reform around the 4 new sets of chromosomes. Cytokinesis has divided the cells into 4 new daughter cells that are all different haploid gametes with 23 chromosomes in each. In summary, at the end of meiosis 4, 4 daughter cells are created, which is different from mitosis, in which only 2 daughter cells are created. The 4 daughter cells for meiosis now have half the number of chromosomes and 1/4 of the number of DNA than the original cell.